Hi, my name is Emily Sheppard, and the title of my poster is Evaluating Climate Change, Improving Survival Rates of Chinook Salmon in the Pacific Northwest. For my capstone, I work with NOAA Northwest Fisheries, where I focus on life cycle model research and specifically Chinook salmon life stages in response to climate change. Roughly 40% of salmon populations have been extrapolated from their natural areas due to climate drivers, which can include increased temperature, sediment, food supply, flow, and others, and all can have a negative effect on salmon survival at all life stages. Therefore, life cycle models are used to study how different life stages, for example, egg, fry, small, or adult, are affected by certain drivers, as depicted in Figure 1. The aim of this project was to evaluate the vulnerability of life stages in relation to climate drivers and find mitigation strategies to increase survival. For my research questions, my first research question was which salmon environment functional relationships are most influential to life cycle model research? And that led to my second research question, which was what actions can be taken to increase survival rates of Chinook salmon? As mentioned before, I interned with NOAA Northwest Fisheries, where we focused on identifying established salmon environment relationships and gathering them into a central user-friendly library. The goal of this project was to improve efficiency, transparency, and communication about the information driving life cycle models and help influence management decisions. My internship consisted of conducting literature reviews of life cycle models, qualitative interviews where we conducted 10 informational interviews with scientists discussing the most important salmon environment relationships, then created a GitHub data repository that acted as our easy access library to provide researchers with important information for life cycle modeling research. Looking over at my results section regarding my first research question on the most influential salmon environment functional relationships, the results from the interviews demonstrated that the egg to fry life stage of response to temperature was the most influential salmon environment relationship in modeling research. Figure 2 demonstrates that the egg to fry life stage, according to researchers, was the most important life stage. Of the 8 researchers that we interviewed, 6 of the 10 said that egg to fry was the most important salmon life stage, and all the researchers agreed that temperature was the most influential and studied environmental factor in response to life cycle model research and development. For this reason, I chose to research how increasingly warming temperatures are going to affect egg to fry survival of Chinook salmon in the Pacific Northwest. And Figure 3 demonstrates that all the environmental factors affecting life stage, temperature was the most important factor. The results of research question 1 led me to research question 2 that by mitigating a plan to decrease temperature in freshwater spawning habitats will increase survival in the egg to fry life stage. Therefore, I focus on a 2-stage mitigation strategy you can see in Figure 4, starting with a proposed policy plan that will implement restoration actions leading to a decrease in temperature and an increase in survival. Policy would include local governments or US EPA to introduce laws to reduce impacts of climate change, including restoration, mitigation for overfishing, protected spawning areas. Restoration would improve stream flow, reestablish habitat, and increase shade cover by planting trees in riparian zones. The key takeaways from my research, through qualitative interviews and literature reviews, I found that egg to fry life stage in response to temperature change is the most researched and influential life stage in life cycle model research, as eggs have the lowest temperature tolerance and variability, with thermal tolerances on average 8 degrees Celsius lower than other life stages. Thus, this is a key way to predicting how fish populations will respond to climate change by understanding the mechanisms of thermal tolerance in embryos. This leads to policies which should focus on restoration actions to decrease temperature in spawning areas, and when these actions are implemented, will aid in the recovery of Chinook salmon populations in the Pacific Northwest. Further implications of my research can include restoration work in freshwater habitats to protect established riparian zones, which can include planting native trees and plants to increase shade cover and lower temperatures. For policies, local and national governments should introduce policies to increase stream flow by limiting water withdrawals, removing barriers to fish movement, which can lead to significant reductions in direct and indirect mortality from mainstream dams, including restoration, for example, of the Lower Snake River through dam breaching and protection of riparian forests. Furthermore, this will require additional funding, which can be used to support research that can inform management decisions. Finally, by introducing public engagement to increase awareness and involvement in climate-related issues and increase voting participation, this will lead to further action against climate change. These implications focus on egg fry survival in relation to increasing temperature. The end goal will be to stabilize and increase Chinook salmon populations in the Pacific Northwest as we prepare for a warming climate. Thank you for listening.